Hello everyone, welcome to the next video lecture on the topic NGS analysis. In this video lecture we will be learning how to remove some of the genomic contaminants and RNA contaminants that are present within your FastQ files. In the previous video I learned how to remove the adapter sequence using a FastP command, right? So in this video lecture we will be using a tool called as Bowtie to remove some of the genomic contaminants that might be present within your FastQ file. So based on the article that you learned, basically the sequence read is used for assembling the Pseudomonas aeruginosa sequence, right? So afterwards they will check for SNPs. So in this case they have used a rat model in this case, which is an animal model. So might be a case that when genomic DNA was isolated or strain was isolated from the industrial leakage, some amount of rat genome might also be present within the particular sample. So that rat genome reads will also be present in it. So we need to remove it so that only we could get the Pseudomonas aeruginosa in this case. And along with that, chances are that there will be some RNA sequences that might be coming out in the genome sequence analysis in this case. So we also want to remove that. And also, because as humans are handling this particular sequencing data, chances are that human sequences will also be present within the sequencing data. So we also want to remove that. And apart from that, some additional sequence that might be present is that the microbiome other microbial genome sequence that be present there, because there will be a microbiome that is present within industrial fluid, right? So that is an optional case also, and also some bacteriophage genome sequence, because Pseudomonas is a bacteria, so bacteriophage virus will be present there that might be infecting that also. So that's also an additional case. But in this video lecture we will be removing the human genome sequences, the rat genome, and also the RNA sequences or ribosomal RNA sequences that may be present within the FastQ file for the Pseudomonas. So what I will do is that first I need to download the genome sequences, right? So this is the NCBI database. I will just click here, that is the genome. I will select the genome and I will type in human. So this will give it a human reference genome file from different strain. So this is the reference genome which has the tick mark in this case. So I will just click on it. So this will open up this particular data. So you can see different information here. 3.1 GB of genome size is there. The GC content percentage, it is from chromosomal level, and yeah, so all this information is here. So you can just go through it. So in this I will just click on this FTP, and this gives you a list of genome assembly structures that are present here. So you can see that there is a coding sequence file that is present here, the protein sequence, the RNA sequence, and the translated coding sequence file that is present here. So from that I need to get the genomic sequence. So I will just go to it, I will right click it on it, and then I will just click on copy link. Then I will just go to the Ubuntu terminal. I will open up the folder that I am working in, and using the wget command I will download this particular file from the URL that is just provided. So this is how you download a human genome or any other genome data that is present here. And already in my system I have already downloaded it out. So I will just list it out. You can see that the first folder here, that so this is the file that I have just downloaded. Similarly, I also downloaded the rat genome file from the NCBI using wget also. And apart from that, I also downloaded the rRNA sequences from a particular database called as Silva. So I will show you how. So this is a particular Silva database. Is actually a particular database that consists of 4 ribosomal RNA that is found in all the organisms. So we can download the FASTA files of ribosomal RNA that is found within all the organisms. So what I will do is that I will go to this option download. I will click on RK. So this option opens up this particular list. So this I will click the current, and afterwards the export, and finally it will open a file tool which is which consists of a list of all the long large subunit ribosomal RNA and small subunit ribosomal RNA FASTA sequence and all. So I want to download this particular sequence. For the large subunit one, I want to download this particular subunit one, that is LSURF taxa silva.fasta.gs. So I will click on right click on it. Sorry. So I will just right click on it. I will copy the link, and using the wget function, I will just copy paste out similarly to download the particular file, and then press enter. similarly afterwards I also will download the small subunit RNA. So you can see that the file is there, 77 MB is there. So I will just right click on it, copy link, and similarly what I have done earlier, I will go to the Ubuntu terminal, I will just type in wget and then copy paste link, and then what you say, press enter and download the file out. So in this case I have these 4 files, right? So it has been done. So in the next case what I want to do is that I want to decompress this file. This is the format of a compressed file, .gzip file. So I want to unzip it out. So what I will do is that I will just type it this particular option, gunzip. Then I can directly copy paste this particular sequence. So I can directly do that. I can decompress this file. But what happens is that it decompresses the file and creates a folder within this particular folder. Inside that folder, the files will be present. But I want to have only the files within this particular analysis folder that I am currently working on. So what I will do is that I will type add a code -c to it. So it actually decompresses this file, and the file that is present within this folder, right, that will appear within this analysis folder itself rather than creating a new folder. And also I don't want this particular name. It's kind of like hard to remember. So what I will do is that I will change this name into using this particular format .faster, or I will just add human genome. Okay. So what it happens is that it unzips this particular folder. It creates a folder by this name, but as I have given this extra sequence or format here, right, so this particular name will be changed to human genome.fasta. So I will just press enter of this particular command. So I will just wait for some time. So basically, as you can see, I have downloaded the file and compressed them out. So there are 4 different files for the human genome and from the rat genome, and for the large subunit I have given this particular file universal su.fasta, and for the small subunit universal su.fasta from these files. So next we want to like combine these FASTA files into a single FASTA file. For that we will use a command called as concatenate or cat. Then I will type in this particular FASTA file. Okay, so I have typed in this particular four folders, the files that are present here, and then I will give in the final FASTA file name contaminants dot FASTA. So what it does it that it actually combine all these file names that are given here using the cat file, and then it gives out this particular file. It combines all these sequences that are present within the FASTA file into a single file under the name contaminants.fasta. So I will just click enter. It will take some time. So basically it is done. You can see that from all the files that are present here has been combined into a single contaminant.fasta file. So I will just check it out. I will just use the head command to print the first 100 lines of that particular file that is being generated. You can see that there are files that are present here. So based on this data, it has been evident that the contaminant.fasta files have been generated. So what we want to do next is that then download a particular tool called as Bowtie2, which is used as an indexing tool to remove the contaminants that are present in a FASTA file. So I will just clear this out. I will just use the command sudo apt install Bowtie2. So as you can see, this tool has also already been installed in my laptop, so I prefer that you install it in yours also, and then I will call up the particular tool. So what I will do is that I will type in the command Bowtie2 build contaminants dot fasta. Okay, contaminant index. So what it does is that it opens up the command for Bowtie2. It builds a index file, that is contaminant index file, from the FastQ contaminant file that we just have. So basically, if you just press enter, it will actually start up, and it will take some time. Basically, it's not like 1 or 2 hours it will complete. For me, it took like 8 to 9 hours just to build the contaminant file. So I will just wait for some time until the build option has been completed. So basically the contaminant files are now built. So we can actually use this particular index files, contaminant index files, to remove the any contaminant reads that are present within the FastQ file. So for that, what I will type is that I will type this Bowtie2 -x contaminant index. This one, the first FastQ file, that is SRR396 is the first file, and then I will also type in the second one, and then type in unconditional, and then for the output name I will type in clean reads dot FastQ, and then I also need a stat file, so I will type in aligned SAM, the format of SAM file I will. So what this called, that it calls out the Bowtie2 command and the contaminant index file that you have generated, these 6 files, and we have also given the forward and reverse read here, and it goes through this forward and reverse read. The index file goes through the forward and reverse read, removes the reads that are similar to what is found within this index file in the FastQ file, and returns the output in the name of clean reads 1, both as a paired end file. And it also gives up the stat file in the format align.sam. So .sam is also a particular format that will be using for the next generation sequence analysis and all, but in later on we will learn what that particularly means. So I will just press enter and wait for some time until the work has been done. So as you can see, the files have been generated. Basically there is not much reads that are getting aligned in this case. Some of the reads like 13 or 51 reads actually exactly aligned to particular files, but it doesn't make up much of the percentage in this case, so that the first file has less contamination in this case. So I will just list it out. So I have got the stat file here. I have also got the output file after contamination have been removed, that is forward file clean reads.1 and clean reads.2. So using FastQC, I will just convert this particular file into a readable format. I will just type in both of their names, so it will just start the analysis work and all. Okay. Okay, so they have been generated. I will just type in explorer.execute. I will just open this one out. I will just open the second one out also. Okay, as you can see, there is not much change in GC content because the percentage of contaminants was also quite less here. But basically, it is an innate property of Pseudomonas genome here, as we have earlier went through. Yeah, okay, so basically in this lecture we have learned how to remove genome contamination from the FastQ file using a tool called as Bowtie2, and also how to download some of the FASTA sequences from NCBI and databases. So I hope you have understood this particular lecture video, and we will see you in the next lecture soon. So thank you.